Roshan, thank you so much for the opportunity to share what my team has been doing. Um, and it was really, I guess, a bit of a retrospective. Today, there's going to be tons of information, kind of a bird's eye view of things going on, kind of like drinking from a fire hose. But I decided to title it The Perspective from the Field to the Forest and Back Again. Okay. Um, I'm going to just briefly tell you about Ag Research Service and then give the three main examples from macadamia to ohia to coffee. But first and foremost, it is not a small team. The teamwork with the agencies, the universities, the amazing faces you see here, team members that I've had the pleasure and honor of working with in my laboratory. The current team, Lionel, Blaine, Caitlin, Melissa, Marianne, Xiaohua, Megan, all of this really is to their great work. You heard PBARC, you hear a lot of acronyms, the government. We are the Pacific Basin Ag Research Center. We're Ag Research is the research arm of the United States Department of Ag. So while PBARC is in Hilo, we serve the entire state with a mission to develop sustainable ag systems and pest management programs. So we are here to support the state, the Pacific Basin, and actually all areas of the country and the world. I'm part of the Tropical Plant Genetic Resources and Disease Research Unit. I'm one of 5 scientists and a curator with the Germplasm Repository. So Roshan kind of gave my, yes, I started in 2002. Hard to believe. I have this giant title of what do I work on, which sounded like holy crow. To try to summarize it, when it comes to pathogen detection and management, we're trying to figure out what is it, how bad is it, and how can we manage it. And once I started, a lot of these crops were new. The experience of some things, I figured, well, how many things could there be? Well, unfortunately, there's a lot. Working with corky bark on things like langsap, looking at orchid diseases, characterizing scabby canker on guava, trying to help manage long gone fungal problems, being part of a pipeline for short and long-term solutions of bacterial blight of anthuriums, and even being a part of the first field trial of the Bovaria bassiana to control CBB when it came in 2010. I probably could talk a million years, but I won't. I will try to give the highlights of the first field, Phytophthora species attacking macadamia in Hawaii. So when it comes to, and I'm going to, again, recognize the OGs of Dr. Nagao, Dr. Ko, Dr. Nishijima, Alan Yamaguchi had the honor and privilege of working with these pathologists when I first started. When it came to macadamia quick decline, there was this persistent problem since 1986, killing productive trees in a commercial growing area around Hilo. There were a number of suspects, a number of fungi that were suspected to cause MQD. And in 2005, in a field in YK Ag Experiment Station, there was high frequency of MQD, which allowed us to go into the field, collect samples, actually identify the prime suspect of Phytophthora tropicalis. And I like to say that these fungal pathogens silently kill trees from the inside out, and they often go unnoticed until it's too late. So once we knew what we were dealing with and we could define the symptoms and signs, we wanted to see how can we control it. And we started with laboratory tests, as we often do, looking at what was registered for use against some of these pathogens in Hawaii for macadamia, and seeing that this chemical is probably could do something. It did something on plates for the fungi. And then we went into designing a leaf bioassay. So not only to understand the different variety responses, but when we're looking at, well, how can we screen quickly in the lab or the greenhouse before doing tons of work in the field? And so some of the variety testing, and then also developing the leaf assay to look at the fungicide effectiveness before we got into the field. And right away, we knew there was good potential with this product called phosphite. And so setting up this giant study, we looked at ways to apply, being the drench or this passive injection. Hopefully you can see my pointer. Retrofitted this way of injecting, which took hours, and switching to a system, an active system that could get the product in quickly, and marking where we would see the symptoms and signs, following disease progression over time, comparing the treatments, collecting data. It's no easy task. And again, we thank the team. And so not only did we then figure out how to use a leaf bioassay to screen varieties, look at fungicides. We also determined that you could drench or use tree injections to arrest progression of symptomology of MQD in the field, actually eliminate visible symptoms, and prolong production, which is great for the farmers. And we thought, okay, good. MQD, good. Well, believe it or not, less than a decade later, there started to be a re-emerging problem of MQD, and going in the same sleuthing way we do things, we found another species of Phytophthora causing macadamia quick decline. Now, outwardly, it looked very similar. Some of the differences was more autumnal leaf litter, but definitely a different pathogen. This is the spores of one versus the spores of another. And at the same time, UH actually had a macadamia variety trial that had gone in the early 90s over in the Kainaliu station. And this was an opportunity to, in a way, see real time what was happening with infection. And so we took a lot of data collectively over there, determined that it was in fact Phytophthora heaviae causing MQD, and looked at a number of varieties. So right away could tell potentially ones that fared well and ones that were very susceptible. In the end, the variety trial, a whole lot of trees actually died. The university needed the land for something else, but it was a good opportunity to gather all of this information. Okay, fast forward now another few years with, for the first time, finding Phytophthora cinnamomum in a field down in Pahala. Now, cinnamomum actually is the Phytophthora species causing the most problems in macadamia elsewhere around the world. It had been found once in very low incidence, maybe the 80s. We had never seen it again until we did now. And then we realized that a lot of this, well, this slow decline, canker, dieback. Sometimes it looked like MQD. Sometimes it looked like it should be cinnamomum. What was going on? We needed to try to figure out what species was causing all these problems, and this increased incidence and severity, why now? Okay, so to do some improved methods of identification, detecting it in the field, we went about, well, how best to sample? I mean, we thought we were doing pretty well, but could there be improvements? And so as opposed to deep sampling, well, we figured out we could do more shallow sampling and actually pick up Phytophthora quite readily when we concentrated around wounded areas and signs of trunk bleeding. Then we developed a way to, okay, it's one thing you find the DNA in the tree, but is the pathogen still viable? That's always the question. Do you need to manage it? So making improvements instead of using a whole apple to bait out, which is typical for what researchers do. My postdoc, Chris Ferenc, actually figured out how to use apple puree. So we could do this almost field-friendly test with baiting, putting small samples that you could collect in the field, and definitely improving our success rate, figuring out how to find it, how to figure out if it was viable, and figure out then what species was causing MQD. So phosphite had been long used. We're excited that over time there's new chemistries to look at. And this is just showing you the types of assays. If here on the left, control, these are what these different fungi look like growing on the plate with no control. And now you look at different compounds and are these products fungistatic, meaning if the pressure of these chemicals are removed, will the fungus grow again versus things that will kill it, fungicidal. So we're happy to report we are finding some good things that hopefully the industry can utilize soon. Okay, varieties. Well, of course, all commercial varieties currently being grown were highly susceptible to MQD. Well, as trees needed to be replaced in the field, what good varieties are there? What good rootstock and what good varieties for production? And so we have an extensive program for screening, and we moved on from the small leaf to actually using these little seedlings. And so not only looking at how the pathogen infects the leaf material, but roots, which is typical for Phytophthora, and what does it do to the stems? And so perfected this type of assay. And we also, so you had two MQD species of Phytophthora, also Cinnamomes present, Palmivera. These things are multi-host pathogens. So we wanted to see, well, how can we protect against multiple things in the field? Okay, and this is just how you take data, how you break it down. Caitlin Bronco really helping lead this phenomenal work to really just take tons of data. Where is Phytophthora infecting? What parts? Where can we recover the pathogen from? And then to summarize, well, when you're looking at percent incidence of disease, is there a preferred tissue type of these pathogens? And how do these different species infect the different varieties? And I know, like I said, it's like, oh, too much to look at and stare at, but definitely can answer questions or provide more info in the future. But what we're starting to see is that different varieties are more susceptible or resistant to certain species. And so this will be great information to know what growers can replant with hopes of some definite tolerance or resistance. Okay, we also do potted plant assays to understand once infection occurs, how long does symptoms take to develop? Where are the signs and symptoms? And to get into some newer, advanced technology. Can we detect things early? Can we find a spectral signature? Could we use a drone for early detection in the field? So you could use those products that can get into the tree in the place they need to, so they can be effective. So combating Phytophthora, attacking macadamia, and this is largely in a way the summary for lots of what we work with. There's never a silver bullet. You always need to use an integrated pest management approach, having short and long-term strategies. Of course, regular field scouting, early detection and control is key. Maintaining plant and soil health, hygiene and sanitation, removing that inoculum source from fields. As when it comes to Phytophthora, it needs good drainage. You want to prevent wounding. And so some of this newer work with new varieties and potentially new fungicides, of course, we want to minimize tree loss and maximize production for our growers. Okay. Whew, deep breath. That was the field. Okay, now into the forest, switching to Rapidohea death, which I'm sure probably many of you have heard of and are well aware of. Again, the team. I remember the call in 2014. With the original ROD squad, Dr. J.B. Friday, who's on here, Dr. Flint Hughes, many of you we've worked with over these years. It turned into the tiny team to essentially worldwide collaboration, and can't thank enough for the passion and commitment of the variety of people that we work with. Ohia, known as Metrosideros polymorpha, part of the Myrtaceae family. It's incredible. It really is the tree of life for everything it does culturally, environmentally, ecologically. You can't say enough about the what this does for life. I was amazed. It's the first thing you see in the lava field, and it's old succession growth. You can find it pretty much everywhere. Here's the map of where Ohia is located throughout the state. Located on all islands, the bulk of it being on Hawaii Island. It's beautiful, it's colorful, the varieties, it's many forms, hence polymorpha. This kind of summarizes the picture, healthy forest down in Puna, 2008, and what it started looking like 4 years apart. Now, there had been forest issues before with Ohia, attributed to cohort senescence in the 70s and 80s. There's rust, there are foliar pathogens and canker pathogens, and even drought and volcano issues, but never before had we seen something like this. First discovered then in 2014 with this rapid wilting of the canopy. We would hear time and time again people saying, "Oh, I'm looking at these trees; they look alive, and in no time they just rapidly die." Once we got inside, you could see the signs of the pathogen here, this dark discoloration of the wood, and we found the fungal pathogen Ceratocystis. And I say not one, but two. And this is where the story maybe even got more complicated. So for pathologists, it's critical. It's one thing to constantly find a pathogen, but you have to prove it's actually causing the disease. You see what is happening in the field. You recreate it actually with healthy seedlings and the sole organism. And this is what was the first key. Going through seeing how long it would take, rapid Ohia death. It actually took months. We didn't see any signs until suddenly symptomology appeared. We saw the wilting, internal discoloration, re-isolate the pathogen, and could now say that Ceratocystis was in fact killing trees. Okay, so in order to see, well, it's one thing again, it's there. Is it viable? Things would take weeks, even a month, to get the viability assays. So the first key, Dr. Wade Heller, postdoc working on it to create a rapid detection test that you could take any kind of sample. There's actually two versions, an enhanced sensitivity test, even for environmental samples through soil or water even. Using qPCR, you could now detect one or two of the Ceratocystis pathogens, always having a control to know the test worked. And we currently here still are testing statewide samples submitted to by all of our partners, which is really the first step towards efficient management, knowing what's there. And this test HCOA uses to enable interstate transport due to an imposed quarantine. Okay, I say the name evolution. Well, when we found it, we thought it was Ceratocystis fimbriata, which they cause tree diseases around the world. We then found two. We were calling it A and B, doing tons of work with experts and consulting with cultural practitioners and understanding what Ceratocystis was here and making sure it was not the same thing that had just come to a new host. But in 2018, for the first time, fungal pathogens were given Hawaiian names to understand what was going on. Ceratocystis leuco ohia, Ceratocystis huli ohia, and these were the two new species involved in rapid ohia death. Well, calling it ROD, that was kind of the umbrella, rapid ohia death. Two pathogens, of course, science-wise, they're two different diseases. Dr. Mark Hughes did a lot of work with this, along with others, to figure out leuco ohia actually is a vascular wilt pathogen. It can move in the vascular system, the spores. It colonizes quickly. It causes widespread mortality in the canopy. Ceratocystis huliohea is actually a canker pathogen. So what's tricky, you look at this side on the left, the outward symptomology, it looks pretty similar. You start to see inside, there's very different signs and symptoms in a way. But they both ultimately result in the death of ohia. Okay, so I mentioned these ceratocystis, they are sometimes broad host range pathogens, and we have a lot of agricultural crops and other things growing here that other parts of the world, there are ceratocystis attacking them. So we wanted to see, well, what else could be vulnerable, especially growing very close to ohia. And so trying to get our hands on all material, things we work with. You can see the huge list, inoculating with the rod pathogens, and fortunately seeing no effect. So as of very recently, we thought these two ceratocystis species only host was ohia. Hot off the press last year, we found out collectively through a study with experts in Brazil and New Zealand who were screening kiwi fruit germplasm for a ceratocystis outbreak that happened in Brazil. We sent the strains to see, and sure enough, this was the first time leuko-ohia actually infected something else. Okay. Understanding how these pathogens infect all of the goal of management strategies. This tries to summarize it. Pictures again, the expert, Dr. Jenny Juswick, and others, figuring out, Dr. Kyle Roy doing a lot of work, ambrosia beetles and the frass and sawdust and what can serve as inoculum. How can we treat wood that's infected? Okay, so it could be utilized. Different phytosanitary methods, dip diffusion and kiln heating and vacuum steam. And is there, it's virtually impossible once trees are infected to cure. So could we protect trees by treating them before infection? That was an ongoing study. Of course, not applicable for forest use, but you can see just the studies to try to manage rod. Another kind of initial groundbreaking, there are many varieties and species of polymorpha throughout the state. And so were all of them going to be as vulnerable to rapid ohia death infection? And so Blaine Louise did his master's thesis with us, actually doing the first screening to see what would happen. And right away, we would see that there was variability in how ohia would respond to infection, with definitely very susceptible, but other more tolerant. And so that was kind of the first real glimmer of hope that the forests would be okay. And this was really the springboard for the much larger ongoing resistance program, the ODRP, ohia resistance program, being led by Dr. Mark Hughes and others through Forest Service with lots of support throughout the state. Okay, so if you're not familiar, Metrosideros actually exists many places around the world, and one very similar tree occurs in New Zealand. And this started up a collaboration because, well, what did that mean for the native forests of New Zealand? So working with researchers and others and cultural practitioners, through Better Border Biosecurity, Ag Research, Plant and Food Research, we decided to help screen their New Zealand native Metrosideros species here. So we set up a partnership, and they sent seed sources for Metrosideros excelsa. This is one of the pictures. So with, unfortunately, ohia, with leuko ohia, polymorpha, within a month, you see rapid wilting. Right away, we were seeing, wow, 6 months plus. We do not see the same thing with Excelsa. Very limited when you look in extensive staining versus very limited. So host pathogen interaction, and in this case, it seemed like host could actually compartmentalize and stop infection, which was great. Didn't, I mean, that's kind of an ongoing. We are now working with other species. These are some viney species of Metrosideros, this one in particular, Perforata. And unfortunately, we're starting to see that they can be susceptible to leukohea. So I'm going to kind of end this, the forest section with a broad summary. Well, what's going on in Hawaii? So leukohea and huliohea are found widespread in Hawaii Island. Kauai, Hawaii Island, it's probably like a 90-10 split leukohea. It's kind of a 50-50 split right now on Kauai, and we're, the greater researcher community, we're trying to understand these pathogens in various environments and what that means and what's happening. There was, well, up until recently, one detection of huliohea on Maui that had been eradicated for quite a number of years until just recently. Huliohea has been found in a few more places, and Oahu, from the first finding of huliohea, it's kind of been one or two over the years. This really is to say that the teams on these islands doing detection and management, boy, early detection, and fortunately, it is huliohea alone on Maui and Oahu. Ongoing studies with all of these rod isolates that we detect, grow out, keep as culture collections, work with experts like Dr. Tom Harrington for population genetics to understand how these pathogens change over time. But collectively, everything is with statewide testing to support management strategies, research to support management and conservation. And I'm going to leave it with, of course, it's a team effort. We say AFUFA here, all for one for all, and most ohia forests are still healthy. So please take that away. Okay, another deep breath. We're back to the field. So, ongoing and just ongoing challenges and management. And in 2020, the probably most recent widespread pathogen attack for Hawaii, coffee leaf rust. We're the last coffee growing region of the world to get CLR caused by this obligate parasitic fungus, Himalaya vistatrix, which means it needs a living host to survive. And these are what these spores look like magnified. Again, the teamwork is incredible. I just can't say enough. Totally blessed to be working with the people that we do. CLR first detected on Maui in 2020, and really within 7 to 8 months, it was widely distributed across the state just due to the nature of this pathogen. Involved in many areas of research, from early detection, figuring out the genetics and race, fungicide testing, and I'm going to give the updates on varieties and natural enemies. And you can kind of see that theme throughout, that variety testing is helpful for all host pathogen interactions we work with. We're always looking for something to manage it, whether that be fungicides or potential biocontrol. But it is a complex, every process and every disease is different. And really you have to hone in the IPM strategies for each one. Okay, so to characterize the pathogen for CLR, again, rapid detection using traditional endpoint PCR, and then also working again with Dr. Wade Heller, experts like Dr. Aim at Purdue and others to develop the rapid qPCR assay for for detection of this pathogen. Working with, like I mentioned, the rust expert, Dr. Aim, also others, Dr. Kokbach at Ag Research Service, doing these molecular analysis. Where did it come from? What is it most similar to? How many times was it introduced? And here's where we think it kind of came from: Central America and the Caribbean. One introduction that then spread because there was essentially no genetic diversity back then in the 2020s. Working with the world's experts, Dr. Varzea and his team, who does race identification. So this pathogen is different. Again, it needs the living host to survive, but there's many races, and that, yeah, I could talk just forever on that. Races and varieties and how they interact and what race can attack what variety. Well, we needed to know what race we have. So, unfortunately, all of the varieties of coffee currently grown here commercially are all super susceptible. So, to know what race, now we know what varieties to look at for potential resistance. And so working with them and their set of differential plants, that they're essentially the one team that does this in the world with all of these plants to figure out. We have race 24 here in Hawaii, which contains these virulence genes 2, 4, and 5, which means any variety containing these corresponding genes are susceptible. So things with Kona typica, Bourbon, Katuai, Katura, grown throughout the state, highly susceptible. So we want to avoid introduction of new races because there's probably at least 55 plus. We also want to avoid the evolution of new races because of what this pathogen can quickly do over time. Okay, so to screen varieties, how do we do that as rapidly as possible with as much material that we can get our hands on? Looking at these pictures from potted plants, small seedlings, things in the field. Well, in large part, Blaine Louise, back from Rod, working with coffee, optimizing this leaf disc assay. So you can do it screening as fast as we could, ramping up as much as possible to prioritize the varieties for additional testing. And this was in large part how to screen a population that had been with Dr. Chifumi Nagai on Oahu, thinking ahead decades and decades before CLR even came to the state, had a breeding program crossing resistant varieties with the farm, each island's farm special varieties to try to create resistant varieties that farmers wanted that were growing here. But because CLR had not been present, and it's virtually impossible if you send plant material away, the challenges of bringing it back. So all of this material was on Oahu, but untested. So this partnership developed, partners at HARC, Dr. Ming Lee, and others, getting material, leaf material through the proper channels to us at the laboratory to set up leaf assays, to ramp up to test this material as quickly as possible. Okay, this is the summary of ongoing looking at 788 different crosses of these individuals, resistant crossed with Hawaiian cultivars, and the large percentage of resistant material here in the state. And so this is to show you what this leaf disc assays looks like and what CLR with spots and copious amount of spores looks like on susceptible varieties, which, as I mentioned, sadly, most are in the state. But under high concentrations, resistant or tolerant material you can see in this instance in the middle, well, I don't see spores. Well, that's because it doesn't produce any. So although you might see some chlorotic spots, the pathogen is not actually completing its life cycle by producing more spores. So it gets in, it infects, but it actually doesn't continue to produce the next inoculum source. And then there are many instances, as you can see from this material, resistant. So this is how much, we put on these leaves, but you actually see nothing, which was phenomenally awesome. And when you see how much of the resistant material, and you look at, well, this bar here shows that almost all of the resistant material, there's no spore production even, okay? And this is kind of a higher concentration than what you would see in the field. Excuse me. So this was great news. This unbelievable material is present. Okay. So other things that we've been working with other partners, there we noticed, sadly, this is a very heavily hit CLR field. This is the leaf material and leaf disc assay to show you. This is essentially a defoliated tree, heavily infected with coffee leaf rust. This is a control. Well, there were others in growers' fields that were actually surviving quite well in areas where most weren't. So the collection and testing of this material on established farms. In one case, this is called after a grower's child, Maite. Doing all of this to actually show it is true that it is resistant material existing in the field. And with the partnership initiated into tissue culture, Steve Starnes, UH Hilo, now it's with HARC to further multiply this material. It's been genotyped by one of the world experts, Dr. Dapeng Zhang, ARS in Beltsville, understanding the genetics behind it, and it cups well. So this is, the best case scenario for having material available pretty quickly for growers. Okay. Also, UHC-TAR has what looked like after a number of seasons, resistant material in the field. And so with partnering with Andrea and Matt, doing these types of assays to show, once again, susceptible controls, but the material in the fields are resistant or highly tolerant. Okay, in this case, maybe we saw a little bit of chlorotic specks, but no spore production. And for the most part, then all of these results in the laboratory are correlating to what we see in the field. There's a lot of great material. This proves then that I should say more resistant material is growing and available, and they've been propagating, and growers can actually purchase this material. Okay, I'm going to end. I'm sorry, I know I'm going long. One of my favorite subjects, fungi attacking fungi. So we noticed right away that there were things growing on the spores of CLR very early once CLR was in the fields. And this extensive study, thanks to Lionel and Blaine and Brian, doing a huge isolation of what these potential mycoparasites are, ID them, again, working with experts to figure out their species, a variety of locations, and now doing assays to see, well, what happens if you have these biological control fungi in the presence when infection occurs? Okay, so we could study these and better understand what's happening by infecting spores. Okay, so that's what we're looking at here to see the mycoparasite efficacy. If we have spores and you apply these potential mycoparasites, do they colonize these spores? And what are they doing? Are they using them as a food source? We're trying to figure this out, but you figure we have, 100 plus isolates to look at, and we want to find the best ones for the most practical application in the field. Okay, what if, because a lot of the protectant fungicides need to get there in the field before infection occurs? Well, what about using potential microparasites as that role? If you get that onto leaf material to protect it from being infected in the first place, what happens? And we can use these leaf disc assays to show again that there's the control, lots of spore production. Okay, this is water control. We don't expect to see spores. Well, what happened? Well, look at all these are four examples of different genera. Some protect, some you don't even see symptom development. Okay, this is just to kind of show you that we have things that we're looking at not only as suppressive, where if there are spores there, they like to attack the spores, but if they're there on clean plant material and the CLL spores land, they actually, they're not going to infect. So there's a lot of work ahead, but we're hopeful that this could serve as a valuable tool for our coffee farmers as part of this IPM plan. Okay. Um, ongoing, doing all this extensive work trying to understand coffee leaf rust in the field, we actually found another emerging disease. Different leaf spots did look different than anthracnose or cercospora spot, and it turns out here is another pathogen attacking coffee as a leaf spotter just recently, and unfortunately, it really can wreak havoc in the greenhouse. Now, this is nothing new. Other parts of the world have it, but sadly, now Hawaii does too. Okay, constantly on the lookout. So with invasive species, there are many detrimental diseases elsewhere that we do not want here. And here are some of them. So, constantly be on the lookout of symptoms that look suspicious or different than what you expect. Okay, I'm going to end my group. I don't know if they love it or not, but I love Scooby-Doo, and we always say, hey, we're like Scooby-Doo, plant pathology, that these pathogens, I would have gotten away with it too if you weren't for these meddling kids. Okay, we're these meddling kids we're ripping off the masks of the villains. Of course, prevention rather than cure. Holistically, right? It's one thing to have it, but can we delay the onset of symptoms or reduce infection levels or cut down on? So you may not have to eradicate, but can it be economically feasible to work with what's there, but still be able to produce? Short and long-term solutions, it's always key to understand, well, what are you working with? Like we said, we thought MQD version 1, and then you find version 2. Things may look the same symptom-wise, but it can be a different pathogen, and they can actually respond differently to fungicides. So one becomes ineffective. Well, maybe that's because you actually have a different pathogen. Everything we're doing, we're trying to be as environmentally friendly, economical, practical for our growers. We want something that can be used. And with that, we can all take a deep breath. I want to say thank you very much. Again, it's an honor and privilege to do what we do with all of these people. Many of you I know are on this call as well. So thanks. And with that, I'll try to answer any questions you may have.